Welcome back, everyone, to the last session of the day of the Genome Application Symposium. So our last session will be on temporal genomic analysis and historical sampling, and this session will be chaired by Tereza Manousaki and again by Maria José Ruiz Lopes. So go ahead, the floor is yours.
Good afternoon, everyone, and thank you for being here. We are going to, sorry. So welcome to this last session of the day, Temporal Genomic Analysis and Historical Sampling, that will be led, as José said, by me. I work at Doñana Biological Station, and Tereza Manousaki, who works at the HMCR. I hope I said that all right. So temporal genomic analysis and historical sampling. So temporal genomic analysis involves comparing different time points to understand what changes have occurred to our populations, in our case, from a genomic perspective. This temporal genomic analysis can now, thanks to the advancing technology, expand thousands of years, and we go from ancient DNA that's typically found in the records and fossils, historical DNA, and the most clear example comes from museums, and contemporary DNA, which is the younger, newer DNA, and we can compare it through time series. These changes and this time frame comes also with a strong change in DNA integrity, which is one of the major challenges we can find when comparing historical samples, that ancient DNA is highly degraded. The integrity seems to be better, or should be better, if you have preserved your samples well, as you come closer to the present. So temporal genomic analysis and historical sample have been used to estimate different population parameters. Here are some of the most important ones. So estimating changes in genetic diversity, like genetic erosion, changes in connectivity, or changes in population size, all of great importance for conservation. They also allow us to identify adaptation, either to the environment, and climate change, or specific stressors. There are some challenges and considerations that are important to take into account when we are studying, designing a study on that includes different time points. And here we are mentioning some of them, but after this little talk, we'll use, as everybody else before, a Mentimeter, where we are going to want to go into more depth about what are your challenges and what you consider when you are designing a time series study. So first, because it's a time series, how many points do we need to sample? That is going to depend a lot of our question. What is my species generation time? Because we have to consider that to understand those changes. What time would give an appropriate baseline? Not all the points in the past are going to be an appropriate baseline, depending on what we are studying. Are historical samples, museum samples adequate for our question? Can we use an opportunistic approach, or we need a long-term or planned study where we are doing like sampling time points in populations closer to the present? And regarding sampling, it is important to make sure that we are sampling the same populations at each time point. So it is very important to consider whether we have population structure, what sample size do we need, or are historical samples adequate, would be other of the common questions that we frequently come across when we are designing our temporal and historical sampling studies. So again, as we are going to talk a little bit about a couple of case studies, I'm going to be very brief because I already explained mine this morning. So I'm going to focus on the study of pesticide-resistant development in mosquitoes. So vector-borne diseases are growing, and we are focusing on Culex species that are common in Europe. They are vectors for multiple diseases, including West Nile virus, which is this disease that I talked a little bit about this morning for the ones that were here, that is transmitted between different Culex mosquitoes to birds, and occasionally can affect humans and horses and cause severe disease and even death. So we know that there are two main species in our area in South Spain that are transmitting West Nile virus, that are Culex perexiguus and Culex pipiens. And we have known since the early 2000s that West Nile virus was a problem here in Spain, but the cases were really rare and spaced. So we had cases in 2004, 2006, 2010. So there wasn't a systematic vector control program tackling mosquito abundance. However, in 2020, we had the first big West Nile virus outbreak with 77 cases, all in the south of Spain. And in that time point, we started stronger vector control programs and the use of pesticides, in particular targeting adults, and sometimes also larvae. Since then, the situation in Spain has changed dramatically, and we now have had cases of West Nile virus every year. And this year, well, when I did this slide, it was 80 cases, but we are over 100 right now. And pesticide use is becoming more and more common to tackle the problem of mosquito abundance, especially around rice fields. So how does mosquito pesticide resistance evolve? It can evolve through standing variation, through a standing variation or de novo mutation. So we have, imagine that we have an ancestral population. If a resistance mutation can emerge, but it might not be associated with the pesticide, then it becomes present at low frequency. And when the pesticide is introduced, then those resistant genotypes are selected, and then mutations are at high frequency in the population. When it is created de novo, as a result of the pesticide that is introduced, we have the resistant genotype selected after the mutation is introduced after the pesticide, and then we have resistance mutations at high frequency with reduced diversity at link loci. So for this case study, we have been sampling populations in these locations since 2010. So we have multiple time points that we can compare to see how resistance has emerged, studying both selected sweeps and also comparing population parameters across time. So now I'm going to give the floor to Tereza, who is going to talk about her project that tackles multiple topics, from seascape genomics to population, adaptive population.
Okay, so I share my screen. Can you see? Is it okay?
Yeah.
Yes.
Okay. So yeah, I want to say before that we have 90 people at the last hour of this symposium, which is amazing. I will also give you a short overview of my case study, where we are dealing with temporal data, very recent, from very recent in terms of the time scale that Marie showed. So in our case study, we work on the population genomics, including all the adaptation genomics and seascape genomics that Marie was mentioning now, on an endangered sponge species, Spongia officinalis. So Spongia officinalis is the classic bath sponge that we have been selling across the Eastern Mediterranean for since antiquity. It's an iconic species. It has been exploited since the ancient times, and it has quite high socioeconomical importance. There are islands that were basing their economy on that particular species. So it's endemic to the Mediterranean. It has higher abundance in the eastern part of it. We have the sponge grounds, and there's a decrease from east to west. which may reflect the different conditions within the sea, like salinity, temperature gradients, and so on. So it is highly threatened, and it has also so great socioeconomic value. However, we know so few about these unconventional strange species. However, there has been one major PhD from my colleague Thanos Delianis, with whom we have both designed this, which is the genetic analysis of this particular species using microsatellites in a study 15 years ago, which is where I have access to all their samples. So he did an extensive sampling. He has sampled about 500 samples across the Greek seas that he has studied with microsatellites. So what we are doing now in our case study is to do a deep search analysis in the evolution of this species across the Greek seas, Aegean, maybe also Ionian, using whole genome resequencing data, so genome-wide data this time. And we want to examine the distribution, the abundance of the populations, the patterns of genetic diversity, the demographic history, the adaptive potential, the seascape genomics, so the relationship with environmental variables and probably the response to the climate change that is really intense in this region, and set the ground for future restoration efforts. So that's what we are aiming at doing. We are targeting to sample different sites within the country. We have already sampled a great part of it, so we're in the middle of the sampling currently in the case study. And this is what I want to show you at this particular session, which is what we got from getting DNA from those old tissues. So we had those old tissues. You can see those are the first efforts. So Xenia Saropoulou, I will talk to her at the end, is a PhD student who is actually working on this. And we have two samples, sponge samples that are from the 15-year-old study. And those are three newly sampled individuals. So you can see the difference at first that we got from the DNA extraction. So at the beginning, we were really disappointed, but then Xenia worked hard and she worked on different protocols and different practices, and she did manage to get what we consider good quality DNA from the old samples as well, but it was really a challenge. So I'll also show you some of our sampling pictures because they are really beautiful, that we had last April at the south of Crete. So what we aim to do is to define neutral and adaptive genetic variation, to study the genetic diversity, the genetic structure, and demographic history. Putting the temporal scale of on this, we want to see if there are newly specimens of the same regions as 15 years ago with the same allele frequencies. If we have some populations lost, some haplotypes even. We don't know what you were saying now, Marie, that you need to know the generation time, and so we know nothing about the species. We want to see also those parameters to see if we have offspring and how maybe the genetic structure has changed within those 15, 20 years that the samples have been taken. So this is the team of the case study. Thanos that I mentioned already, Xenia, who's a PhD student, Emanuela and Grigoris were heavily helping us with the diving, because this is really difficult. And with that, I finish with my case study. And I would like to trigger discussion with you so we can launch our Mentimeter, which is my first Mentimeter, and I hope it will work. So how do now I have to stop sharing to share my Mentimeter, right?
Yes.
Okay. No.
And what was the code?
I took out the slide. So I will, yeah, thank you. So you get the Menti code in the chat?
Mhm.
Okay. Share, and then I present. No? What can you see? Can you see okay, I see
Yeah, it's
replies.
okay.
Yes, yes,
Okay.
it's working.
So our question is, how many of you have been working with old tissues or old DNAs? And what is the age? So there are some people that have really old samples, some really, really old, 50 to 150. We have some that are over 150 years old. Okay, it's really what we were looking for in this session is to actually get feedback from you, that you have all this experience from the different samples, from sold samples, especially people working in museum samples. So most of them have some old samples, like in our case study, which are also already a pain from our experience. We had also some failures in the library prep afterwards, I have to reveal to you, that we need to deal with. So they are really unconventional, and the risk is at all different levels, because you expect to have good DNA, then you go to the sequencing center and everything is fine. But it could be that's because they are also unconventional species that we're discussing in the previous sections. So we have people that are working on really old samples. We're really looking forward to get your feedback and your experience. Should I move on to the rest? To the next? Okay. So where are they preserved, your samples that you're dealing with? Yeah, ethanol. Dried skins sounds difficult also. So I guess most of the people are, of course, in ethanol. And also we have silica.
Silica, dried skin, so that's from museums. So,
Yes, Marie?
no, I was wondering, I don't remember like the next question we had, but I was wondering like if people have worked with different type of preservation methods, which one work better for them? Guess it depends on the age as well, but.
Yeah, given from the responses, which would be the most easy scenario and which would be the most difficult scenario? To me, ethanol, but I'm a bioinformatician, so I will not even try to respond. But who had really hard times from the response? And I guess it's also a matter of, yeah, José.
I can just I was the one that wrote dried skins because we work with some museum samples that are from, I don't know, from 50 to 100 years old, or even a little bit older. And the main issue there is that we often don't know exactly what kind of treatment the museum specimens went through. So we suppose that they are dry, but they could have had very different products applied. And age is obviously one thing that is related with the DNA integrity. But then from our experience, we see a lot of heterogeneity even with age. And so I think it also has to do with the way it was preserved, even if we classify all as dried skins. What we see is that we never had a major issue. Well, DNA extraction, of course, is an issue because DNA is degraded. But if it is degraded already in the tissue, there's not much you can do about it. And then the amounts of DNA is another difficulty because, of course, degraded DNA, old DNA would come in lower concentrations, in lower amounts. And so normally for library prep, this requires PCR-based techniques. It's hard to apply PCR-free library prepping to these old samples because of the low amounts of DNA. And this, of course, being forced to use PCR-based libraries, it will come with all the uneven representation along the genome, PCR duplicates, and all that. There is also DNA damage that we normally take into account. This can be treated during the treatment of the processing of the samples using user enzyme, which corrects the deaminated uracils, which are normally the problem of working with old samples. There is also a possibility of working with that bioinformatically later, as ancient DNA is often done, but this means losing some variants because we need to remove all variants that can be due to damage, and so it ends up removing variants. Then the consequence of have short inserts, short DNA fragments is that when we have very short inserts in the libraries, and so when we're sequencing, we end up having redundant sequencing, of course, because paired end sequencing is shotgun sequencing is the norm now, and so we're essentially sequencing the same fragment twice. We sequence adapters, so we have to have in mind in the processing all those issues. And of course, we end up having to sequence much more for the same amount of coverage, for example. And so to reach really high coverage, it's a huge effort for sequencing. And so normally what we do is that we couple this low coverage approach that João explained in the last session, but with individually barcoded individuals, so that we can sort out possible bias contribution, individual contribution to the analysis that we're doing. But in the end, well, it all comes also to the question and to actually whether we will have the power to infer the differences of the things that we want to separate in terms of temporal timescale. But we've been quite successful. It is more expensive, but we try to overcome that by employing more lower coverage analysis, compensating with higher numbers of individuals, etc., etc. But it works. It works. And for temporal analysis, I think temporal analysis. I think they are really important because when we're working only with contemporary samples, we're missing many processes that we're trying to infer what happened, and this way we have a window to the past, bearing in mind all the situations that Mary also explained before.
So
Wow.
Gabor is saying that silica gel work for him well in the case of some animals. And he's also asking about any experience with DNA degradation in the preservative. We have experienced DNA fragmentation in a stored material, which goes along the lines that Jose was just talking about.
In order to me, oh, sorry.
Yeah, go ahead,
Sorry.
Howard.
I have a different
Yeah,
question.
So I'm coming from the plant world, and in plants, silica gel is the standard way. But we tried with some butterflies. We also tried with fin clips from fish, and it worked very nicely, and it yielded very, very good DNA comparable to ethanol. And in some cases, ethanol is very difficult to transport and carry. Therefore, silica gel is a good alternative in some animals. On the other hand, for example, in beetles, we had to drop all the beetles collected in silica gel into the bin because all the DNA was fully degraded. So for beetles, it doesn't work. And for the other one is we have this experience when everything started to degrade, although stored properly, and especially I have a feeling it's something something related to genome size, because very large genome plants DNA started to fragment easier. So probably it's something related to that.
I guess they also fragment easily in every pipetting you're doing if it's much bigger genome size in every treatment. I wanted to ask José about the SNPs that you get in the actual, when you reach the coverage and you do all those bioinformatic analysis, do you see fewer SNPs compared to modern samples? I mean, do you see the effect of all those layers that you put in your effort to accomplish it?
Yeah, well, yes, applying more filters will end up losing some variation, but the studies that we've done were actually whole genome scans of association studies, and it works very well, which means that the density of SNPs that remain is dense enough to be able to, of course, then it depends on the recombination landscapes along the genome and the sizes of the populations, all that. But we do the same with more modern samples, and I don't see much difference in terms of density of SNPs.
Wow. So I move to the next question then.
Yes.
So what is the biggest challenge in obtaining high quality historical DNA samples? We have some examples here. If you have other ideas, you can put them in the chat.
Mm-hmm. Katya is saying that people sometimes did not preserve their old DNA tissue samples after finishing their study. So thanks, Katya, for the input.
Maybe one challenge is also to have metadata. I'm moving to the next.
What tools do you use to overcome these challenges? Like pulling samples or what Jose was saying before, your bioinformatic approach.
Yeah, collaboration with people working on ancient DNA.
Hmm.
Lower coverage because it has reduced biases. How would that work?
Mapping that reference.
Yeah, mapping to a reference. I guess for contamination. Good bioinformatics.
Pre-screening and running pilot sequencing tests.
So the levels of complexity are really higher.
Yeah. So there are common questions like screening samples to assess endogenous DNA that to avoid like or minimize contamination issues. Adapted laboratory. I don't know what that exactly means. Maybe modify the library prep.
You mean the spiking?
No, there is an answer called adapted laboratory, which I might
Yes.
mean.
Protocols. Okay, that means that you need to fine tune.
Conditions and protocols. I couldn't see it.
Yeah, you cannot just take a protocol and run it. Okay. Then I move to the next.
Mhm.
And that is what type of questions would you like to answer with your old samples, with your old data? Yeah, changes in genetic diversity.
There are changes in genetic diversity. Genetic basis of adaptation represented in museum collections. We can't sample. Assessment of diversity that we can't sample anymore. To use as a baseline. Demographic changes. Evolution of effective exercise.
It's what José was saying, like having a glimpse in the past. So when you're trying to infer, you also have data from there. Study extinct populations, things that don't exist anymore. Okay. extinct populations, yes. So what would be the main problems, the main limitations of your temporal genomic analysis, if we rank them?
Temporal genomic analysis in general, yeah.
Yeah, I guess difficulty of high quality DNA.
So it seems that the common major limitation is like the quality of the samples. That seems to be the major limitation when we are talking about temporal genomic analysis. That it goes hand in hand with incomplete historical records, because where does the samples come from, etc., is often or might be a problem. Small sample sizes, unbiased and available data, and limited time span follow a little bit behind, but the major limitation seems to be like adapting our protocols to working with samples that are not as good as we would hope.
Yeah, I can imagine how difficult it is to find something that really works as is on your samples. José?
Yes, just a note on this. Of course, for the past, we can't control what has been collected and how it is preserved and what it represents. But we also need to consider that the present is the past of the future, right? And so, coming back also to Katya's comment in the chat, preserving samples right now, so biobanking, metadata collection, is really important, so that in the future, that brings us actual temporal analyses are sort of more like monitoring studies. We can, in the future, maybe not us, maybe the next generation can go back in time from properly preserved samples and will overcome some of these difficulties if we have in mind that it is important to make the most out of biological collections that are being done right now. That's just my comment.
You would be really positive if this happened. Okay, we just said that historical records will be improved because we will do it better. Imagine even that even the statistics of sampling many individuals from different populations in the past, when people didn't know how much we would use them in the future, so now. So I can imagine how difficult it is to find 20 individuals from population that has been sampled 100 years ago. Okay, and this is our most important question. What is the biggest challenge in integrating old samples with contemporary data?
The inequality.
Missing analysis methods is a problem in every scientific publication. This is exactly what we're also working now compared to older generations.
Numerical method.
And also accommodating the differences in power in the analysis, that's also really difficult because the low analytical populations will drive the whole analysis down, if I'm expressing this correctly.
So yeah, there are like common topics in all these answers, like metadata missing, analysis methods, and removing batch effect biases and differences in data quality seem to be like the three major challenges to integrate historical and contemporary genomic data.
Confirming how important all these are. Okay. And I think this is our last question. And that is, if you were a reviewer, if it would be acceptable to have the same sequencing coverage in the old samples with a new one, or you would accept having much lower coverage or even extremely low for very important old samples. Yeah, I guess there's so many challenges that it really makes sense, but the target would be to deal with those challenges and be able to get better sequencing coverage also in the historical samples. There is one very optimistic person.
Two.
More. And with this, I think we're closing also this session. It was really interesting to see how challenging it is and how much progress we can do on that part. So unconventional and historical samples, I guess, are really the hardest to work on. So I will stop sharing and thank you all for being here so late after so many hours in the morning.
Yes, thank you, everyone. I think it was a really great first half of the Genome Application Symposium. Thank you all for being here, for the nice discussions. And tomorrow we will start at 9:15 CEST. And thank you all, especially the chairs, for running these discussions. Thank you all.
Thank you.
Thank you for the
Thank you
organization.
all. Bye-bye.